Hello, my name is Danique Beijer and I'm a researcher at the University of Antwerp in Belgium. And I will be presenting you some of the results from our recent study titled Nonsense Mutations in alpha-II-spectrin in 3 families with juvenile-onset hereditary motor neuropathy. Hereditary motor neuropathies are a rare form of inherited peripheral neuropathy, and HMN are characterized by a slowly progressive distal muscle weakness and atrophy in the limbs. Depending on the HMN subtype, it can be accompanied by other characteristics like pyramidal tract, vocal cord, and diaphragm involvement. The increased implementation of next-generation sequencing in the last decade has allowed for the discovery of many novel genes involved in inherited peripheral neuropathy. And similarly, our study made use of next-generation sequencing in a large cohort of 73 families with hereditary motor neuropathies. In this study, we report on the findings of 3 of these families, in which we identified heterozygous nonsense mutations in the SPTAN1 gene encoding the alpha-II-spectrin protein. Spectrin is a component of the cytoskeleton, and it is abundantly present in both the peripheral and the central nervous system. However, its precise role in human disease remains largely unknown. In this case, we used either whole exome or whole genome sequencing in 2 or more individuals per family, and we identified a nonsense mutation in SPTAN1 in all the 3 families. The use of Sanger sequencing allowed us to confirm an autosomal dominant segregation together with the peripheral neuropathy phenotype. As a next step, we used patient-derived lymphoblasts to assess the effect of the mutation on mRNA and on protein level. And by culturing the lymphoblasts in normal conditions and upon blocking of nonsense-mediated decay, we observed that the mutant mRNA is in fact broken down by NMD. Protein extraction from these lymphoblasts with subsequent Western blotting showed that the reduced mRNA levels also result in reduced protein levels in individuals carrying an SPTAN1 nonsense mutation. These findings suggest that loss of alpha-II-spectrin expression is the cause for hereditary motor neuropathy observed in these families, and the identification of SPTAN1 as a novel cause for hereditary motor neuropathies is actually quite intriguing, as previously mutations in SPTAN1 were associated with a spectrum of epilepsy phenotypes. The most recent report was actually on 2 families with SPTAN1 mutations and a complex neurodevelopmental phenotype. So the identification of these 3 novel mutations in hereditary motor neuropathy broadens the phenotypic spectrum of SPTAN1 associated disorders. And it firmly implicates that loss of function alleles in alpha-II-spectrin are a cause of motor axon degeneration in the peripheral nervous system. I thank you for listening, and for further details I'd like to refer you to our manuscript.